Thank you very much. Thank you, Michael, for the very kind invitation. My name is Christos Pliotas. I'm a group leader at the University of Manchester, and today I'm going to present to you our recent study published a month ago in Nature Communications from our group, in which we used SMA extensively for purification of. So, a big background of my research. I don't know where this keeps coming. So we have been working on membrane proteins. A long-standing interest of the group is mechanosensitive ion channels from bacteria. Recently, through collaborations and from our own group, we have extended in our studies in other membrane proteins, including ion pumps like membrane paraphosphatases and peptide transmembrane transporters like the SBMA. Also human potassium ion channels and TRPC ion 5 human channels, and also the outer membrane protein complex bank. But in the interest of time today, I'm going to discuss this project, as I mentioned earlier on, which was published recently. So we are a highly integrative structural biology group. We have focused on the EPR methodology, which I'm going to briefly touch on and explain to you what we can do. And then the idea is that we monitor conformational ensembles of membrane proteins, and the most interesting or novel states we then pursue by cryo-EM. We have expertise in our group, or X-ray crystallography to solve these high-resolution structures. We do a lot of computational work as well, in which we employ molecular dynamic simulations to restrain the distances that we obtain from this main methodology. And we also do a lot of electrophysiology to characterize especially the ion channels. And I'm going to show a little bit on this as well, along with some native mass spectrometry we've done in SMALPs. So the method involves the acquisition of interspin distances. We routinely measure between 20 to 80 angstroms. The world record is around 160, 170 angstroms. And you can determine oligomerization, conformation of membrane proteins in lipids. It has really high resolution. You can also use DNA binding or substrate or drug binding if you spin label your ligand. And you can also do time resolved PPR with millisecond resolution if you flash, mix, and freeze your sample. So these are the facilities of my laboratory in Manchester. I arrived in Manchester 2.5 years ago, and we got hold of state-of-the-art pulsed EPR spectrometer, Q-band. This is the most highly sensitive commercially available EPR spectrometer. We also recently, through a Canadian-based company called HiQ, we obtained the first quantum EPR spectrometer in Europe and in the UK, which can only take 3.5 microliters of sample. And we independently have developed a flash mixing freezing instrument to be able to enable in-cell time-resolved EPR. So we introduce we modify cysteines. We introduce cysteines on our proteins, and then we modify them with MTSSL spin label, which looks like that. And then what we end up with is with a very high resolution nanoruler, in which allows us to measure distances between different spin label sites. So if you have, for instance, a pentamer, you would expect to get 2 distances due to pentagon symmetry. If you have a heptamer, then you get 3. If you have a dimer, you expect to have 1 distance. And if you have, for instance, a GPCR which is monomeric, you can either introduce 2 spin labels on 1 monomer and study intramolecular distances and conformational changes within the GPCR, or introduce 1 spin label, and then you can address oligomerization with 2 GPCRs come together. And over the years, a lot of different spin labels have been developed by different groups in order to be able to incorporate these paramagnetic species on proteins and perform EPR. And EPR has certain advantages, like, for instance, the size of the label is of the size of an amino acid, so it's less disruptive compared, for instance, to single molecule FRET or FRET that they use these bigger fluorophores. Also, you don't require donor acceptor molecules, only 1 label does it all. And in the case of membrane proteins, it's quite favorable technique because you can do any kind of lipid bilayer system that you can think of, because unlike cryo-EM, for instance, that you have to freeze the sample on a grid, here you just freeze in a tube. So you can do, for instance, liposomes. You can do nanodisk with the MSP apolipoprotein. You can do saposins and also SMALPs. And the talk today will focus on SMALPs and also bicells. The resolution of the method. This is a structure that we obtained several years ago, that we have the 7 spin labels on the mechanosensitive channels in the transmembrane domain. And as you can see here, these are the distance distributions we're getting from the EPR methodology. The resolution is really good. So it's 25.9 angstroms was from spin to spin distances on the structure directly. And what we obtained from the mean distance of the distribution for the first distance between subunit A and subunit B was 25.9 angstroms. And with the relative ratio of these peaks as well, you can also determine the oligomeric state of the protein based on some basic trigonometry, obviously. So what is the pipeline? How we use this technique? We start by selecting the model from the AlphaFold or cryo-EM structure or X-ray structures initial model. Then we explore the conformational space by molecular dynamic simulations. Then we create this kind of spaghetti containing multiple frames of the protein moving in space. Then we obviously do our basics and wet lab work. We perform the distance measurements, and then we use these distance restraints to go back and inform our molecular dynamic simulations. And then, for instance, out of 1,000 frames, we fit the data and select for the frames that are present without the ensemble. And we can also determine the proportional representation of each frame within that ensemble. So that's what we really do. We combine molecular dynamic simulations with high-resolution distance restraints and obtain entire conformational ensembles of membrane proteins in lipids. So this brings me to my talk today, the main test case from our lab. So over the years, we have been working, we had a long interest in mechanosensitive ion channels, and this time we endeavored to study on the human potassium mechanosensitive ion channel. So this kind of problem has not been made possible by cryo-EM and crystallography over the last 15 years. So the TRAAK potassium mechanosensitive channels is expressed in the nervous system and regulates rest and memory potential, and also act as a mechanosensor for heat, stress, and fatty acids. It's also very important for neuroprotection and axial potential conduction. I don't know if you have ever rubbed your eyes and you saw flashes of light. This means that you're without any light entering your eyes. This means that your TRAAK is not working properly, actually. So as I said, it can be activated by pH, it can be activated by tension, by heat, by polyunsaturated fatty acids. So what was the case here? In 2012, Roderick McKinnon from Rockefeller University reported an unswapped state of the TRAAK channel. And 2 years later, they reported a structure of the swap state. So the only difference of these two channels is this, if you can see here, this extracellular, very unique for K2P channels, 2-port domain channels, domain called the CAP. And as you can see, the difference is that you have monomer 1, monomer 2, and they're like stuck together in this case, whereas here they're sort of perplexed, right? They're swapped, as we call it. And the only difference is in these residues on the very top. So what happened is, over the next 14, 15 years since then, a lot of leading groups around the globe, they have started obtaining novel, new cryo-EM structures over the years. But what happened is, in all K2P channels, 70 structures were in this state, and only 2 were in this state. So the question was, is this state biologically relevant? Does it exist in a lipid membrane content? So for that, we wanted to do a full-length human TRAAK. All the X-ray and cryo-EM constructs used for all these structural studies, they have truncated the C-terminus domain, which interacts with cytoskeleton, limiting function. Also, we wanted to do innative lipid membranes, and for this purpose, we used detergent-free SMA methodology. And we wanted to do an unbiased monitoring. What does this mean? Most of these structures, the vast majority, they have used state-locking antibodies. We would not want to induce any bias. We wanted to monitor unbiasedly, entirely unbiasedly, the entire sample and see what is out there. So if we spin label now one subunit with conventional EPR, you will see that there is no distance separation. So then we reach a dead end, as had happened as well with all the structural studies over the year. However, if we manage to heterologously spin label one subunit with 2 labels, and the other one will be cis-free and then the spin label will not be accessible to the subunit. Therefore, it will be EPR silent. Then the separation of distances we get between the swapped and the non-swapped state and the swapped state is 17 angstroms. And as I showed you earlier on with the resolution we can achieve, this is very, we cannot get really definitive conclusions. But this is not easy. So again, I show you here conventional distance measurement, you will see no change. However, if you heterologously express and spin label the two subunits, then the distances differ substantially, and this is what we try to do. And for this purpose, SMA was instrumental. So what we've done, we co-expressed two constructs, one with two systems, one with cis-free and different tags. Then we obtained two kinds of species, one with two labels, one with no labels, EPR silent. Then three isoforms were expressed, right? A tetramer that is unwanted. We don't want that because it's going to mess up things for us. The EPR silent construct that is there, but we don't see by EPR because EPR detects only paramagnetic species and the system of interest. Then we used SMALPs and we broke the membranes down and we trapped all the native lipids from the parental membrane used for cell expression. And we got nice disc, SMA discs with native lipids of the three species. And then going through a nickel column, this is V-tagged, obviously, so it went to the flow through, and then we end up with this sample, this is our sample, this is EPR silent, so we don't see, and this is the interest within a SMALP disc. And then we just, since this discs, this protein reconstituted in SMALP discs contained spin labels at the desired sites, we performed distance measurements using EPR spectroscopy. And what we found out was, and this was striking for us at the time because we didn't know what we were going to get actually, was that both states coexist in lipid membranes, in cell parental membrane proteins. And also we even quantified the ensemble, and we found that the dominant species, around 60%, was the swap state. And this is the state that the vast majority, overwhelming majority of cryo X-ray structures have been solved in. So this explains all these structures that have been obtained by multiple world-leading groups over the last decade and even longer than that. The other interesting thing was, okay, now we monitored the ensemble, if you like, and we saw that both species are present in membranes. Is there any trigger that can activate them? Then we screened different conditions, because we didn't know if it's biologically relevant. Now we show that it exists in membrane, so it should be biologically relevant. But the question is, what is the trigger that can shift that equilibrium? And after testing multiple conditions, again with EPR spectroscopy, we found out that temperature affects that equilibrium. And it's well known over 10 years or 20 years ago that TRAAK is activated at fever high temperatures around 40 degrees of Celsius. So that was really interesting. And this is something now we're going to move forward and explore in the future to understand why temperature changes the swapped state to non-swapped and why this is happening and what's the biological relevance of this phenomenon. On the side, we did mass spectrometry in which we extracted the lipids that they were trapped and selected by TRAAK before solubilization by SMA, obviously. And we detected really nice lipids, including signaling lipids, which we then tested by electrophysiology. And for instance, for the PI lipids, we saw activation of the channels. For the PIP2 signaling lipids, we saw activation. For the PE lipid, we also detected in our lipid analysis, we saw inhibition, and for PS, for instance, seems to be structural, we saw no effect. So this is a really nice other advantage that SMALP provides us, that we could get the immediate native lipid environment of the channel, and then we can take these lipids individually to electrophysiology and test them. What was really interesting here to say is that PC, phosphatidylcholine, is the most dominant lipid in plasma membranes. The interesting thing here is TRAAK excludes, we detect no PC lipids. So TRAAK, for some reason we don't know, obviously, excludes PC lipids from its vicinity despite being the most highly populated lipid in the plasma membrane that we expressed TRAAK in. And I will close now. I think I'm on time. I will close with my group and acknowledgements. This is my group. You see they're exhausted, but I think they're all happy. And we have, especially Mary, who was the first author in this study, and also Kesho Wahid, who has done all the molecular dynamic simulations. There is a lot of modeling in this paper as well. feel free to read. And the collaborator as well, Bella Boda, who helped with EPR measurements as well in St. Andrews and Manchester. Thank you very much for your attention.
Christos, thank you so much for a wonderful talk. Really, really amazing work that you and your team are doing. Let's open the floor for questions. I'd also want to say thank you for being so wonderfully on time.
Thank
We
you.
have a few questions from Michael in the chat. Do you want to go ahead and ask those, Michael?
Let me find the chat one.
Sure. While you're
so
looking,
yes.
If anyone has any questions, you have precedence over me, but
right.
I thought I'd just pop in a few questions until you get going.
That's a very good question, Michael. I didn't have the time to really go into very deep into the technique in the interest of time, but just to say here that EPR is an ensemble technique, unlike RIME. So what happens based on your first question? It says, do both swapped and non-swapped appear on cryo-EM grids? So the situation here is when you put your sample on a grid and then you start doing the auto picking and then you start the refinement, you exclude, right? You exclude particles along the way. You start with 2 million particles, and then you end up with 100,000, 50,000, 10,000 particles to derive your ensemble and your conformation. During these processes, you bias, right? You ask the software to go for high resolution, and you exclude. Yes, some of these particles might be dirt, might be ice, might be something of unknown nature. But how about having other lowly populated transient states or disordered states there? So you exclude them.
Okay, thank you.
Along the way. Is cholesterol present in the complex? We express this human ion channel in Pichia pastoris, which the equivalent for is ergosterol. We did not detect ergosterol. No, we did not. How can people access a facility? Because it's not if I sort of own or I have this spectrometer through collaborative basis. So we don't charge, we just collaborate and we do science. So you come with a question, really. That's the whole point. You come with a question. And if it's interesting and we discuss and we're all happy, we just move on.
Great. Thank you so much. Any other questions from the crowd? I'm also wondering about polymers. Do you have any favorite polymers or new polymer properties that you'd like to see?
I would like polymers that can accommodate larger. So I don't know if you noticed in my first slide when I showed we have a variety of structurally diverse different channels and transporters. I would like to have something that is bigger, like 150, 200 angstroms, so I can incorporate larger heptameric complexes, for instance, that can span 200 angstroms in diameter. That will be something that I would be interested in optimizing, pursuing, or whatever.
Great. I'd like to ask, Giselle, do you have any polymers that generate large disks?
Yeah, we're actually working on a new development. So we're incorporating a component of steric stabilization, particularly for particle homogeneity. It's still under development, but we have some very hot off the press TEM imaging showing that we're forming these really nice discoidal structures that are around between 20 and 40 nanometers. I have a follow-up question. So if you're keen to try that, we'd be very happy to support you with that.
Absolutely. We're always happy to try on our systems and then tell you if it works. Because even for SMALPs, we tried for this system, we tried DDM, we tried detergents, we tried, all the different kind. And SMALPs work the best. But we have other cases as well within our, the systems we study that SMALPs didn't work and others worked. So really, it's trial and error, and we really have to go the hard way and learn whether it's the best or not the best system for this particular one. Yes, I can see another talk from Evelyn. How did you guys maneuver spin-to-spin interaction in DIR to avoid artifacts? So here is a pulse, right? It's not CW, it's not continuous wave EPR, it's pulse. So we measure direct dipolar coupling, spin-spin interactions. So if you know where you put your spin through side-directed mutagenesis and you covalently attach the spins, then you know exactly where your spins are. And we have structural models, right? We have AlphaFold or cryo-EM structures. So we can model distances on and then derive the differences in the states. I don't know if this answers your question, Evelyn.